Alright everyone, this is the answer key video for the review guide that you guys did this week. The first question is about labeling and identifying monomers and polymers. So this molecule on the left, these pieces are not bonded, so these are the monomers. They're not bonded, they're separated. And then after the reaction, this big polymer, this is the large macromolecule. So remember the pieces, the building blocks, those are the monomers. And then the giant macromolecule, that is the polymer. So you will have to do that on the test. You'll have to circle and label the monomers and the polymers. The next question about the identification of the macromolecule. So notice that there's only carbon, hydrogen, and oxygen. So that means that it has to either be a carbohydrate or a lipid. And this one is a lipid because of the long chains of carbon. There are no rings of carbon, so this is a lipid. Make sure on the test that you're explaining how you know, so don't just write lipid. You need to explain how you know that it is that macromolecule. Same thing for the last question about identifying if it's hydrolysis or dehydration. You need to tell me how you know. So I know that this is dehydration synthesis because we start off with monomers on the left. Notice here water is removed, and then you have a large macromolecule, which is the lipid. Next part about the enzymes. So for these questions, I just looked at where these lines peak, and that is the optimal or the best temperature or pH for that enzyme to work. So look at where the line peaks, follow it down, look at the x-axis. So here are the answers for those. 37 degrees Celsius, which is the temperature of the human body, 1.8, and then 8.1. And for the very last question about denaturation, you guys need to know when enzymes are denatured. So that's when they move out of their optimal temperature or pH. The active site changes shape. When you change the structure of the active site, you're changing the function. So it's all about the shape of the active site. If the active site changes shape, it's not going to be able to bond to its substrate, and therefore that enzyme will lose its function. So again, you need to make sure you explain that on the summative, which will be next week. Mrs. Proctor and I will have our Zooms up Friday today. So if you have any questions, you can pop into Zoom. If we're not in there, shoot us an email and we will get into our Zoom rooms and you can ask us questions. All right, let us know if you have any questions. Have a good weekend.